Hi there, Year 12. Hopefully by now you've received your trial HSC paper back and you have it in front of you so you can look through that as we go through the feedback here and compare your answers to the sample answers and also see where you may have lost some of your marks. This video is going to look at questions 21 to 26. This is the section that myself, Mr. McLennan, that I marked, and Mrs. Litchfield marked questions 27 and to the end of the paper. Let's start with question 21. Now, this question generally was pretty poorly done for what should be a very easy question. And in fact, a lot of students, including the students who got the highest marks, lost some really easy marks on this question. Now, the key here is there's a diagram there, and this part of the question here, the diagram below is one of these practices. And then it's asking you to outline how this practice prevents contamination of the agar plate. So the practice we're looking at here is the lifting of the lid of the Petri dish at a 45-degree angle to then swab on the bacteria or the sample onto the agar plate there. So the answer we were looking for was something about the lifting of the lid at 45 degrees. There were some really great answers that were correct, or true rather, but they weren't the correct answer for this question. So answers about heating the inoculating loop to burn off any bacteria, wiping down the bench, all those sorts of things were things that we do as well when we prepare a sample. But the answer we're looking for, because it relates to this picture here, was something about the Petri dish lid at 45 degrees to stop any microbes in the air landing on the agar plate and to ensure that the only sample that's getting on there is the sample that we want to see. All right, question 21b was worth 4 marks. Compare modes of transmission of infectious disease, including direct contact, indirect contact, and vector transmission. So in this question, you needed to talk about modes of transmission, but you also needed to specifically talk about direct contact, indirect contact, and vector transmission. Now, the most common errors that we saw were students mixing up indirect and direct transmission. So they may have done this by, probably the most common one was talking about sneezing and coughing as being direct transmission. Sneezing and coughing is actually indirect transmission. Direct transmission is touch to touch. If we think about humans, it's human touch to human touch. So things like kissing, things like touching skin, things like sexual intercourse, those are the methods of which there's direct transmission. A cough or a sneeze allows the pathogen to linger in the air, even though it might be in the aerosol droplets, but allows it to linger in the air, and then it requires another person to come through and either walk through or inhale those droplets. So it's not quite direct transmission. That is more an example of indirect transmission. And in that, mixing up those two then meant that some students mixed up their examples that they used as well. Another common example, or common error rather, was not describing fully what each of these things were, particularly vector transmission. Now, vector transmission is when an animal, usually an insect like a mosquito, is able to pass a pathogen from an infected host to an uninfected host through a bite. So not describing that fully was another common error that students made. And also making sure that if the examples you had, that they were appropriate for the type of transmission you were talking about as well. So some really classic examples you could have used. Direct contact could have been something like a fungal disease, or it could have been something like herpes that's transmitted through kissing. Indirect contact, flus, colds, measles, those sorts of things. And vector transmission was more thinking of things like malaria. Now, this is the sample answer from the paper that we marked from. I won't read this out for you, but feel free to pause this and read this and compare it to the answer that you've got in your paper. Now, question 23 was this one about the migration patterns through Micronesia and Polynesia. And it shows a map of Australia out to New Zealand and all the way out to East Island as well. So this question had a couple of key points. One was explain selection pressures, explain gene flow, and explain gene pools, and then link them all together in a coherent sentence. So if we think of our idea scaffold, IDEA, these have been identified for you: selection pressure, gene flow, and gene pool. So you need to describe what they are and then explain how they link together within this scenario here. Now, there were some answers that were fairly creative, and it was good to see you understanding the climate of these areas and how different islands, as we get further south to New Zealand, for example, might present different selection pressures to do with climate. But we wanted to have an example that was related directly to what you could see here in the picture. There was a couple of questions, or a couple of answers rather, that spoke about lactose intolerance and that sort of thing in the diet. And whilst that may be true, that's not something you can deduce or infer from the information you're given here. So again, you needed to relate your answer to this picture and to this information. And in this case, lactose was an appropriate answer for that one. Now, the marking, you need to define gene flow, gene pool, allele, and selection pressures. Describe possible selection pressures on each island for a mark, and then explain how selection pressures impact the gene flow and the gene pool. Now again, this is the sample answer. I'll let you pause this if you like to read this. And we'll move on to the next question. Now the next question here was another one that was generally done fairly poorly. And the reason was, I think, people just didn't read the question properly. This is the really important part of the question. Describe the trends in the graph. Everything they give you before here is just setting up how they did the experiment. What we wanted to see was describing the trend in the graph. Set A, which is the introduced plant species, it loses a lot of mass quickly before it plateaus off. Set B also loses mass quickly before it plateaus off. And the other trend is that set B loses less mass than set A overall after 120 minutes or 2 hours. There was a lot of answers that spoke about native plants having less stomates and being better suited for the environment and all those sorts of things. You have to remember that this question, or the way they set up this experiment, was they blasted a hot fan onto these leaves for 2 hours. And the idea of that was to try and dehydrate the leaves as much as possible, but it's not really replicating a natural environment that we'd expect to see even in Australia in our hottest parts of summer. So describe the trend in the graph. You needed to read that and just describe the trend. Don't try and explain it. Don't try and tell us why set A loses more mass than set B. Just describe the way that that graph looks. All right, question 23 was related to adaptations in plants to maintain water balance. And you're given 3 examples here. Now, normally, when you're given an example like this or a stimulus like this, you're expected to relate your answer to those pictures. But I was fairly liberal in the way I marked that because I know not all of you would have learnt these specific 3. And because we asked for 3 plant adaptations, if you didn't know one of these, I wanted to give you the opportunity to go or relate to one that you did learn or that you did know or feel familiar with. Now, common errors with the answers here. Now, a really common error I saw quite a lot was people talking about CAM photosynthesis and talking about how photosynthesis occurs at night. And what was happening there is you're getting confused with the stomates opening to allow carbon dioxide into the plant at night, which then gets converted to malic acid. But then during the day, the malic acid gets converted back to carbon dioxide when the sun is out to allow photosynthesis to occur. The main difference is the stomates don't need to open. And because the stomates aren't open, we're not losing any water through those stomates in the heat of the day. But photosynthesis itself is still occurring during the day. The sunlight is one of the most important parts of photosynthesis, and obviously there's no sunlight at night. Now, curled leaves and stunken stomates. There were some interesting answers there about absorbing water through the leaves, and the curled leaves pulls the water and drags it into the leaf. And likewise, the sunken stomates mean that the little puddles form in the leaf. None of those are truly what those adaptations are about. Curled leaves and sunken stomates, or curled leaves is really about reducing the amount of surface area that's exposed to the sun. And by having that curled environment, it allows a humid microclimate to form near the leaf. And if there's humidity outside of the leaf that's been formed because of the curled nature, then that loss of water vapor through transpiration is reduced. And likewise, the sunken stomates does a similar sort of thing. It creates a little humid microclimate, which then reduces the amount of water lost through transpiration, the amount of water being pulled out through that transpiration process. And the other thing you need to do with your answer was relate your choices to transpiration. So there were some answers that related to roots and root systems, but they weren't really relating to transpiration, or at least in the answers given, they didn't relate them very well to transpiration. So the best answers clearly describe and explain the link between the adaptation and reduced water loss, and you had 3 examples of that. All right, question 24. Human immune defense against pathogens includes chemical and physical barriers, phagocytosis, and the inflammatory response. Compare plant and human defenses against pathogens. Now, again, this is one where I think students either didn't completely read the question or kind of avoided the parts of the question they didn't want to answer. But here it tells you chemical, physical barriers, phagocytosis, and inflammatory response. So in this question, or in this answer, we want you to talk about all 4 of those things and relate them to human and plant defenses against pathogens. Now, humans are going to have all 4 of these. Plants are going to have chemical and physical barriers, but they're not going to have the phagocytosis or the inflammatory process. So common areas for this question. Most students could describe the human defenses pretty well, although not everyone went into the inflammatory process or the phagocytosis process. Plant defenses generally were not as well answered. A lot of people had the idea that there's a physical barrier such as bark, but the chemical barriers were a little bit weaker overall when talking about those. A few students spoke about phagocytosis inflammation as well. These aren't in plants. There were a couple of answers that said that they were, but the bulk of students that lost marks here lost marks because they didn't address these 2 parts in terms of a comparison. Humans have this, plants don't have that. So again, this is the sample response to give you an idea of what we're expecting in this answer. And again, I'll let you pause this and read that and compare it to your answer. All right, question 25 was the budgie question. I really liked this question, and I was really happy with the way the majority of students answered this question as well. I know with my class we spoke quite a bit about the idea that the HSC, the way it's going, is it's asking questions or asking you to show us familiar skills in unfamiliar situations. And this is definitely one of those questions there, where we're looking at a sex-linked recessive mutation in budgies, which causes them to be yellow. And it's sex-linked recessive. We've done that plenty in lots of examples with Punnett squares with X and Y chromosomes. And the difference here, we're looking at male budgies that actually have 2 ZZ chromosomes. So in humans, males are XY. In budgies, males are ZZ. And females, on the other hand, have the W chromosome. So they're ZW. So again, the skills you need to answer this question are just the same as any sex-linked recessive mutation question with Punnett squares and the like. The difference is the chromosomes here are different, but they give you the chromosomes that you need to talk about. So the question is, if Y is the allele for yellow colouring in budgerigars, identify one possible genotype for a male budgie. So it's not even asking for the genotype of a yellow budgie, just a genotype of any budgie. And it actually tells you here in the question. Male budgies have 2 Z chromosomes, so ZZ is the possible options. Now, the only trick to that is you needed to include the alleles. So I've used Y for yellow, which is what it asks for later in the question. So big Y, so Z big Y, Z big Y, Z big Y, Z little y, or Z little y, Z little y. So they're the 3 options that we're looking for. Now the next question, why is yellow colouring more common in female budgerigars? And this is worth 2 marks. Now the reason is it's recessive, and the females only have 1 Z chromosome. So remembering females are ZW. So because they've only got the 1 Z chromosome, if that Z chromosome happens to have the recessive allele on it, then there's no dominant allele that's going to cover it up. Whereas in males, if one of the chromosomes is the recessive allele and the other chromosome is the dominant allele, then the dominant trait is going to be expressed. In this case, it's not the yellow traits. So females only have the one Z chromosome. They don't have anything that will cover it up. Therefore, the yellow colouring is more common in female budgerigars just purely through the ratio that presents. All right, question 25. A breeder wants to produce only yellow male budgerigars. And he has the following birds: a normal male, a normal female, a carrier male, and a yellow female. So which two birds should he breed together to obtain yellow males? So probably exam stress, but one answer I saw probably a little bit too commonly was the idea of breeding carrier male with carrier male or yellow female with yellow female to produce yellow males. Obviously, males and females don't breed together. We need to have a male and female breed together. Now, in terms of this question here, we want to produce yellow male budgerigars. Males are ZZ, but to have a yellow male, they've got to be recessive Y. So Z little y, Z little y. So the yellow female is going to be Z little y W, and the carrier male is going to be Z big Y, Z little y. These two genotypes here of the normal male and normal female are not going to have that recessive yellow allele on it at all, so we can't breed those together and get a possible yellow male as a result. So these are the only two options that we can possibly breed together. And so the only possible cross that will produce yellow male budgerigars, which is Z little y, is a cross between the carrier male and the yellow female. No other combination will produce a male. Now, here are the parents: Z big Y, Z little y, cross Z little y W. And these are the individual gametes. Here's your Punnett square. And then you can work out the phenotype ratio, where if we have males, this one here, 1 in 4 or 25%, or half of the males rather, 25% of the overall offspring, have a probability of being a yellow male. Now, what we looked for in terms of marking is we wanted you to state what the parents' alleles were. So Z big Y, Z little y for the father, Z little y W for the mother, and then show us a Punnett square and then have some sort of explanation of what was going on there as well. All right, the next question was, in the space provided, draw a table to compare the structure of DNA and the structure of RNA. So this is all about the structure of what DNA and RNA look like. So because it's a table, it's asking you to draw a table. Now, just on that note, there was quite a few students that used tables in other questions, which was great way to present your answer. But in a question like this, where it's asking you specifically to draw a table, then the only possible answer you can provide is one that was within a table. So if you're asked to draw a table, it needs to be a fully enclosed table as well, meaning there's a box around the whole lot, and that's just the format that we expect in the HSC. So if you did that, you got 1 mark, no matter what the rest of your answer was. And then in terms of the answer, the things we're looking for is differences and similarities of DNA and RNA. So DNA is a double helix, RNA is a single strand. DNA has ATCG as the bases, whereas RNA has AUCG as the bases. That sort of answer in a table there would have given you the 2 marks. All right, the next question was relating to our amino acid wheel here. And this table here, or this chart here, is different to ones that you've possibly seen before, but again, familiar skills in an unfamiliar situation. So how you work out the end amino acid, which are the ones on the outside here, is the same as that table that has the AUCG down one side and AUCG down the other side. And the key bit here, what is the sequence of amino acids in the peptide coded for by the following segment of mRNA? You need to remember that mRNA provides the codon, which is the code for the amino acid. There were some students, I think, that converted this to the corresponding tRNA and then worked out the amino acid based on the tRNA sequence, but that was actually incorrect and not what we're looking for this answer. So this is the mRNA code. So all you need to do is CCG, find the C, find the next C, find the G, and the answer there was pro. And here's the answer over here. So pro, GUG, G, U, G gives us val, AGC, so we've got A, G, C gives us Sir, and then UAG here gives us a stop codon. So that was the sequence we were looking for. It was only worth 1 mark, so if you didn't have that sequence, then you didn't get the mark for it. All right, question 26. A mutation occurred in the original DNA strand so that all adenines in the mRNA were replaced by guanine. Using the table from part B, explain the effect of this mutation on the organism. So part B was what we just answered then, which was relating to the amino acid sequence that was produced. So we've replaced the adenines with guanine, and using the table from part B meant that we wanted you to be able to relate the changes to the original strand that you saw. So this was the original strand here. We wanted you to relate the changes to that strand there. So what that meant was, in your answer, what we wanted to see was you somewhere either state the original strand and then state what that new strand would have been, that mutated strand. It was okay if you just had the mutated strand, but somewhere for a mark we were looking for you to replace those A's with the G's to show the mutation that had occurred. Now, the original mRNA produces pro, val, ser, and stop codon. The mutated sequence of mRNA produces the new amino acids, pro, ser, gly, and trip. This codes for gly and trip represent different amino acids, and if we've got different amino acids in the sequence, then that's going to lead to a different protein at the end. So that was 1 mark there. So 1 mark for this section, 1 mark for talking about how the different amino acid sequence produces a different protein. And the final thing, the final mark, was something about having no stop codon and how that might mean the produced protein would be incomplete or a non-functional protein because it didn't have that stop codon telling it to stop producing. All right, the next question, identify a physical or chemical mutagen. My biggest advice for here is just to keep it really simple. Pick something really obvious. It's only worth 1 mark. Radiation is probably the easiest mark you're going to get. UV radiation, gamma radiation, X-ray radiation. I did allow answers that included asbestos and mercury and a couple other chemicals as well. These mutagens are not as clear as radiation mutagens. They seem to have asbestos seems to have some mutagenic effect in certain cells and in certain animals, likewise with mercury. Both of them are carcinogenic, which causes cancer, but they're not necessarily mutagenic. There's a slight difference between the two. So, as I said, I did allow those because they do in some cases seem to cause or have mutagenic properties, but they weren't as obvious an answer as radiation. Remembering mutagens are all about changing that DNA structure rather than causing a chronic irritation like asbestos does, which can lead to something like mesothelioma. Now, probably the most common error for this question was just not reading the question properly and giving a type of mutation as an answer rather than identifying a mutagen. So type of mutation and answer might have been something like lung cancer or mesothelioma, or it might have students might have spoken about a point mutation or a deletion mutation, something like that as well. We really just wanted a mutagen as the answer for 1 mark. All right, this question here, analyze the causes, processes, and effects of chromosomal and point mutations. So this is a question where you need to understand what analyze actually means in terms of the HSC question. So analyze is about identifying components and the relationship between them to draw out and relate implications. So it gives you the scaffold here, the causes, process, and effects of chromosomal and point mutations. So they're the components that have been identified for you. You need to talk about the relationship between them and then relate those implications. So best advice for a question like this, it's worth 6 marks. Plan your answer. Make sure you've got some sort of plan, just a quick couple of dot points on what you want to say, because if you do run out of time, we can look at those dot points and at least see where you were going, and sometimes you can get marks for that. Basically, if it's on the paper, by rights, you are entitled to get marks for that as long as it's relevant to the answer. This is a question that using the idea scaffold would have really helped, and it was really clear those students that had practiced answering with some sort of structure in their answers compared to students that maybe hadn't had as much practice in that area. And so a really common area was just not addressing all the key components, maybe leaving out the causes or the processes or not talking about them or relating them in enough detail. And the other big thing would be to know the difference between a chromosomal and a point mutation, so that you have enough there to talk about. Chromosomal mutation is in the chromosomes. It's passed on to the offspring. An example is Down syndrome. Point mutation is a mutation within the DNA sequence that affects the carrier or the person that has the mutation. It can be a substitution, it can be a frame shift. There are different sorts of processes that can occur. And point mutations, we're normally thinking of things like cystic fibrosis as being one of the most common examples. Now again, here's the sample answer from our marking guide. I split into part 1 and part 2. Part 1 here is looking at point mutations. So again, feel free to pause the video and read this and compare it to your answer. And then sample answer part 2 was looking at the chromosomal mutation. And then this sentence here at the bottom, I just highlighted that because that's the relationship between the two. Both point and chromosomal mutations may lead to changes in the genome. So that's the analysis part of this overall question.